What is the objective of this practical? The objective of this practical is to determine hemoglobin concentration in human blood by Sahli's hemoglobinometer. Now we will understand the principle of this practical. Dilute hydrochloric acid converts hemoglobin into soluble unstable acid called hematine. This acid is brown in color. The color intensity of the solution depends upon the amount of acid, that is hematine, present in solution, which in turn depends on concentration of hemoglobin. The color of the solution is compared with brown tinted glass filter. This is visual method of determination of hemoglobin. Hemoglobin is usually expressed in grams per deciliter of blood. The normal range for hemoglobin for men is 13.5 to 17.5 grams per deciliter, and for women it is 12 to 15.5 grams per deciliter. What are the requirements for this practical? In order to perform this practical, you will need Sahli's hemoglobinometer. It consists of comparator, hemoglobin tube, hemoglobin pipette, and stirrer. You also need decinormal hydrochloric acid solution, Pasteur pipette. It is an ordinary glass dropper with a rubber bulb for transferring liquids, spirit and alcohol swabs, and blood lancet. Now we will understand how to prepare decinormal hydrochloric acid solution. When 36 gram of hydrochloric acid is mixed in 1 liter of distilled water, it gives normal hydrochloric acid solution. Diluting this solution 10 times will give decinormal hydrochloric acid solution. This is how Sahli's hemoglobinometer looks. As you can see, this is comparator. It is a rectangular plastic box with a slot which accommodates the graduated hemoglobin tube. Non-fading standard brown glass filters are provided on either side of the slot for matching the color. As you can see, these are filters. An opaque white glass is fitted behind the slot to provide uniform illumination during color matching. This is hemoglobin tube. The square hemoglobin glass tube is graduated on both sides. One side is graduated in gram percentage from 2 to 22 and other side is graduated in percentage as from 20 to 140. Round tubes are also available. This is hemoglobin pipette. It is a straight glass capillary pipette with only one calibration mark, that is 20 cubic millimeters. There is no bulb in this pipette as no dilution of blood is required. Sahli's hemoglobinometer is also provided with a stirrer. It is a thin glass rod which is used for mixing the blood with the acid solution. What is the procedure of this practical? First, place about 4 to 5 drops of decinormal hydrochloric acid solution in the hemoglobin tube up to 10% mark and set it aside. Then get a finger pricked under aseptic conditions. Wipe away the first 2-3 drops of blood. Then draw blood into the hemoglobin pipette up to the mark 20 microliter. Do not lift the tip of the pipette, otherwise the air will enter the stem. Then carefully wipe the blood sticking to the outside of the pipette tip. Then without any weight, expel the blood into Sahli's hemoglobin tube by immersing the tip to the bottom of the acid solution. Rinse the pipette 3 or 4 times by drawing up and blowing out acid solution. Then mix the contents of the tube quickly but gently by shaking it or by using the glass stirrer. Avoid frothing. Then put the tube back in the comparator and let it stand for about 5 to 10 minutes. During this time, the acid ruptures the red blood cells and hemoglobin is released into the solution. The acid acts on the hemoglobin and converts it into acid hematine, which is brown in color. Then take the tube out of the comparator and add distilled water drop by drop. Stir the mixture each time and check its color against the standard glass. What are the precautions for this practical? First, the finger should not be squeezed as the tissue fluid will dilute the blood. Sufficient time should be given for the conversion of all of the hemoglobin into acid hematine. The color of hematine acid should be uniformly brown throughout the solution. Darker color near the bottom of the tube indicates poor mixing. Color matching should not be delayed as the color of the solution is likely to fade. While matching the color, the graduations should not lie in front as it will affect the color matching. What is the clinical significance of this practical? Hemoglobin is a protein in your red blood cells that carries oxygen to your body's organs and tissues and transports carbon dioxide from your organs and tissues back to your lungs. If hemoglobin level is lower than normal, it means you have a low red blood cell count, which is called anemia. High hemoglobin levels could be indicative of the rare blood disease called polycythemia. It causes the body to make too many red blood cells, causing the blood to be thicker than usual. This can lead to clots, heart attacks, and strokes. It is a serious lifelong condition that can be fatal if it is not treated. Here are your study questions. What are the advantages and disadvantages of Sahli's method for estimating hemoglobin concentration? And what are the other methods for estimating hemoglobin concentration apart from Sahli's method?